This lecture will review mutations. Mutations are changes in the nucleotide sequence. Overall, it is a rare event and almost always deleterious, meaning not good for the organism experiencing the mutation. However, mutations are necessary for evolution to occur, so sometimes they prove advantageous. The mutation can alter the property of a protein that improves the chances of an organism surviving and reproducing to pass on the change. There are known mutagens, both physical and chemical, that increase the rate of mutations from 10 to 100 times. There are different types of mutations. Very large chromosomal alterations or duplications, termed gross mutations, such as the possession of extra chromosome 21 that leads to Down syndrome, won't be the focus of this discussion, although they obviously can have large effects. We will be examining the smaller changes that occur in the nucleotide sequence and the types of effects those changes bring. Point mutations are the changes that occur at one position at one nucleotide along the strand. This can have varying effects, depends on the change. Frameshift mutations typically have a very large effect as they alter all the codons downstream of the change. This slide with a figure from your text illustrates the types of changes that occur when one nucleotide is altered. For example, in panel A, normal gene sequence is shown, and in panel B, one nucleotide was changed. However, this altered nucleotide still encodes a codon for the same original amino acid. Recall that some amino acids have several codons. Thus, this gene changed, but the protein did not. This is a silent mutation. In panel C, a different nucleotide was altered, and now the codon in that position calls for a different amino acid compared to the original. This is termed a missense mutation. Do you think that this one amino acid difference in a protein would matter much? Proteins might be hundreds of amino acids in length. Well, it depends. Sometimes the alteration is tolerated well and the protein still functions quite normally, but this is not always the case. The disease sickle cell anemia has a missense mutation that has altered a single amino acid in the human hemoglobin gene. The result is a serious disease condition for homozygous individuals. In panel D, yet another nucleotide swap has occurred. In this case, the codon that is formed no longer calls for any amino acid. It calls for a stop of translation. This will truncate the protein. This type of mutation is a nonsense mutation. Finally, in panels E and F, two different types of frameshift mutations have occurred. In E, an extra nucleotide was added, and in F, a nucleotide was deleted. In both of these cases, the three nucleotide codon groups are shifted downstream of the change. The entire protein from this point forward will be altered as seen in the new amino acid sequences. This protein will not function at all like the original. This is a major change. You will have lost the original function altogether with this type of mutation. Mutagens increase the rate of mutation. As mentioned, we may have physical mutagens such as ultraviolet light previously discussed. Pyrimidine dimers such as thymine dimers will introduce a mutation at that spot during DNA replication if not repaired. Chemical mutagens can also introduce changes. The following slides are going to look at how these changes occur. A nucleoside analog is a chemical that looks like a nucleotide and may trick a polymerase into using it during replication, but once incorporated does not behave like the normal version of the nucleotide. For example, 5-bromouracil looks like the nucleotide thymine. Once incorporated into a DNA strand, it behaves like cytosine pairing with guanine. This has now introduced a point mutation into that strand of replicated DNA. Intercalating agents are chemicals that insert in between DNA strands. They create a bit of chaos when a polymerase is trying to replicate strands, and often frameshift mutations occur at the site of incorporation. Remember, frameshifts drastically alter proteins downstream of the change. Organisms do have mechanisms to repair mutations. Some mechanisms are shared with a wider variety of organisms. For example, base excision, nucleotide excision, mismatch repair are systems that various organisms can use. They employ a team of enzymes capable of recognizing mismatches, cutting them out, and replacing them. In contrast, the light repair mechanism is directed by an enzyme called photolyase. Only some microorganisms have this enzyme. Visible light wavelengths will stimulate the production of this enzyme by the cell that will identify pyrimidine dimers and fix them. Wouldn't it be nice if our skin cells also had this system, but we don't. Identifying mutants is an important activity for many research scientists. Understanding mutations and their effects are what has enabled science to determine biochemical pathways of metabolism. When an organism can no longer perform a function, finding out what change has occurred allows the researchers to determine the role of various enzymes in a pathway. Of course, there are many other reasons to study mutants. We may want to understand the development of antibiotic resistance or other types of abilities. So let's be sure we understand some terms before we look at these next couple of methods used to study mutant microbes. First, understand that the general population of organisms out there with their normal abilities are called the wild type. When you alter abilities of some cells of that population, you have mutants. Now let's look at three methods that we use when we study mutants. The first method is called positive selection. We use positive selection when the mutant has gained new abilities. The classic example of this condition is the acquisition of antibiotic resistance. The wild type population is not resistant. If some cells acquire that ability, the antimicrobial drug will not affect them. Thus, when you plate the cells on media with the drug, only resistant cells will grow. This is positive selection. Again, you use it to detect cells that have a new ability compared to the wild type. Replica plating is a negative selection method. In this method, you have cells that have lost an ability. For example, they may no longer make their own tryptophan, so they must be supplied that amino acid to grow. When you plate a mixed population of wild type and tryptophan auxotrophs, that's the term for cells that lost an ability compared to wild type. When you've plated that mixed population of wild type and tryptophan auxotrophs, all cells grow on the media with tryptophan in it. You're providing the necessary factor to the mutants, and the wild types can be lazy and not make any for now. However, if you make a replica of the colonies by making an impression with some velvet-like material stamped to the plate and pressed to new media that lacks tryptophan, the mutants will not grow. You have to compare the original plate with the nutrient-lacking plate to find the missing colonies. You'll then verify that those colonies have the trait you desire, and now you have mutants to study. In sum, replica plating, a negative selection method, is used to find cells that have lost an ability compared to the wild type. Finally, let's look at a test that examines if a chemical is a mutagen. To be a carcinogen, a chemical must be a mutagen. However, being a mutagen may not be enough to cause cancer. More testing will be required, but this is a quick, inexpensive screen to detect mutagens and thus potential carcinogens. This is a widely employed test because it's cheap and fast. When you get a promising or a suspicious result, you can verify it with further testing, maybe in other more difficult to grow cells or even animal models. This test is called the Ames test after the scientist who developed it. It requires a Salmonella strain that's an auxotroph for histidine. It can no longer make its own. The wild type can. The reason it can't make histidine is due to a missense mutation in one enzyme in the histidine synthesis pathway. Along with this key bacterial strain, you need media without histidine. You'll need your test chemical, and you need liver enzymes. Liver enzymes are known to sometimes alter harmless chemicals into mutagens, and that's why they're included. You can also run a set without these enzymes. Your negative controls lack the test chemical. They'll include water. You mix the test chemical, the liver enzymes, and the Salmonella mutant together, and then you plate them on the medium lacking histidine. The negative control should show no growth. Remember, the Salmonella can't make its own histidine, so without this amino acid, it can't make protein, and it's not going to grow. However, if the chemical is a mutagen, it may alter the point mutation that caused the Salmonella to be a histidine auxotroph. If it happens to alter it back to the original nucleotide, the Salmonella will now make its own histidine, and it will grow on the media. The presence of colonies on this medium tells you that the chemical is a mutagen. Students typically ask, are there other changes happening in the Salmonella DNA due to the mutagen? Yeah, there are. We can't detect those. We can only detect the mutations that happen to occur in the same spot as the original point mutation. A bacterial culture has millions and millions of cells in it. We only need a change to happen at that spot a few times, and we'll see it as resulting colonies on our plate. So in sum, the Ames test is used to detect chemical mutagens. When set up correctly, it will allow us to visualize a point mutation at a single position in the genome, and we know that the chemical tested caused that mutation. This concludes our section on mutations and mutant detection.